Hello and welcome to another video with Leah. Today I'm going to be talking about Punnett squares and doing some problems in regards to complete dominance using monohybrid and dihybrid crosses, so that you can get a chance to see both kinds of problems. I'm also going to give a brief overview of the terms that I'm going to be using in this video. So for this topic, it's good to know how to do the problems and what the different terms mean. I really hope you enjoyed this video, and if you do, make sure that you subscribe and click the little bell so that you can know when I post new videos. Here we have genotype and phenotype, two important terms that we will be using. So genotype is going to be referring to the genes in that organism, and it can also refer to the alleles in that organism. And alleles are variations of the gene. So for example, if you have a gene that codes for eye color, you might have two alleles. One allele might code for blue eyes, and the other allele might code for brown eyes. Next we have phenotype, and this is going to be the things that we can see, so the observable characteristic. What we can see, what results from that genotype and the organism's interactions with the environment. We have a few more terms here. We have homozygous dominant, homozygous recessive, and heterozygous. And this is talking about the genotype. So what combination of alleles is present in that organism? So for example, if we have two alleles, okay, one is dominant, let's call this H, and the recessive allele is little h. Now, homozygous dominant means that both alleles are going to be dominant. So, capital H. Capital H is going to be the genotype. So, both dominant alleles are present. So, homozygous meaning the same, and dominant meaning the type of allele. So, dominant meaning the capital H, and homozygous meaning that both alleles are going to be dominant. And next we have homozygous recessive. Well, this just means we have two recessive alleles, so little h, little h. So homozygous recessive means that both alleles, okay, are going to be recessive. Next we have heterozygous, and this means that we have one of each allele. So one dominant allele and one recessive allele. So capital H and little h would be heterozygous. And for these problems, we're going to be doing them in regards to complete dominance, meaning that if at least one dominant allele is present, okay, whether it's both of them or one of them, okay, one dominant allele present in that genotype, well, the phenotype is going to be based off of that dominant allele. Okay, and so the other one, the phenotype that corresponds to the recessive allele, will not be shown, okay, if we have at least one dominant allele. So both of these would mean that the dominant allele would be expressed, masking the recessive one and producing whatever that phenotype is. Now, for these problems, it's going to be important to know how to find the genotype of the parent. Okay, and so determine the genotypes of the parents given the following information. The allele for black wings, capital B, is dominant to the allele for brown wings, little b. A bird with brown wings is crossed with a bird with a heterozygous genotype for wing color. So we have two parents. Okay, we'll call the one with brown wings the parent number 1, and the one with a heterozygous genotype the parent number 2. Okay, so let's figure out the genotype for parent number 1. So we're dealing with brown wings. Okay, so the allele for black wings, capital B, is dominant to the allele for brown wings, meaning that brown wings, this little b allele, is going to be the recessive allele. Okay, and the only way that brown wings can be expressed is if we have a homozygous recessive, so little b, little b. So both alleles have to be recessive in order for brown wings to appear in the phenotype. Now we have parent 2, so let's find this genotype. So parent 2, we have a heterozygous genotype, meaning that one allele will be dominant and the other allele will be recessive. So capital B and little b will be the genotype for parent number 2. A gene in plants determines the height. Tall is completely dominant to short. The alleles are labeled as T for tall and little t for short. If a homozygous dominant tall plant is crossed with a homozygous recessive short plant, what is the next generation's ratio of genotypes and phenotypes? Now, when we are doing the next generations, meaning that we have to cross two of the parents and produce offspring, well, those offspring's genotypes will go right in this box. Okay, so there'll be genotype here, 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 and here. Okay, so 4 kinds of genotypes. Now, given the information on the left, we can determine the genotypes of the parents. And those genotypes of the parents, well, they're each going to have 2 alleles. And the genotypes are going to be going in this top area and in this bottom area. So green for one parent and purple for the other parent. And right here, specifically, right where these boxes are going to go 1 allele. So for parent number 1, right, we're going to have, some kind of genotype, and 1 allele is going to go here, and 1 allele is going to go here. Now we're going to have parent number 2. We're going to have 2 alleles for that genotype, and 1 of the alleles is going to go here, and the other allele is going to go here. And we're just talking about the letters. So the letters indicate the allele. So now let's write some notes on this side. So tall is indicated by large T, and short is indicated by little t. And we know that this one's going to be dominant. And now let's find the genotypes of the parents. So one of the parents is homozygous dominant. Okay, so homozygous dominant means that both alleles are dominant. So one parent is big T, big T. The next parent is homozygous recessive, meaning that both alleles are recessive. So the other parent's going to be little t, little t. These are the parents, okay? Now we're going to go ahead and place these alleles around this box. So in this space, we're going to have parent number 1. So let's do parent number 1. Okay, and 1 allele is going to go in each area. So 1 allele and 1 allele. And this makes up the genotype of parent number 1. Let's do parent number 2. Parent number 2 has little t, little t. Okay, and so 1 allele is going to go here, and the other allele is going to go here. And this vertical column is going to be the genotype of the parent number 2. Now let's figure out the genotypes of the offspring, okay? And that's going to be placed in these 4 boxes. So in box number 1, let's do this one. We have to see what meets from the vertical column and what meets from the horizontal row. And so we see that big T and little t meet. So we have big T and we also have a little t. Next we go on to this box, okay? And we do the same thing. What is the vertical part? Well, it's large T. What is the horizontal part? Well, little t. Okay, and so we go on and on. So for this box, we'll have what is the vertical part? Well, it's large T. What is the horizontal part? Well, that's little t. Okay, and so in each one, we're writing 2 alleles. And for the last one, we have big T and we have little t. So now we have found the genotypes of the offspring. Now let's find these ratios. So the genotype ratio, okay, is talking about the genotype, so the alleles, okay, the letters. And so we see here that all of these combinations, this one, this one, this one, and this one, they're all going to be heterozygous. They are made up of 1 dominant allele and 1 recessive allele. Okay, and so the genotype is going to be 100% heterozygous, okay, or large T, little t. Okay. Now let's find the ratio for the phenotype. The phenotype is talking about, well, what's going to happen? If we have this genotype, what's the organism going to look like? Is it going to be tall? Is it going to be short? Okay. So phenotype is talking about what happens, okay, or what can we see? Because genes we can't really see with the naked eye, okay? With phenotype, remember, we're talking about that observable trait. So what do we see in the organism as a result of its genotype and the environment? So for this problem, right, we're talking about complete dominance. And so we know that this sort of genotype is going to produce tall. Okay, it's going to be tall for the phenotype, and all of them are going to be tall. Okay, so 100% are going to be tall. The ratios are capital T, little t is going to be 100% for that genotype, and the phenotype ratio is going to be 100% for tall. Okay, all of them are going to be tall. So let's do another of these problems. This one is again talking about tall plants and short plants. Okay, again, capital T is tall and lowercase t is short. Now we have the point where this differs is the parents. So now we have a homozygous dominant and a heterozygous. So again, let's find the genotypes of these parents. So parent number 1 is homozygous dominant, meaning that both alleles are dominant. And we know that it's going to be capital T, okay, because this is dominant. The next parent is heterozygous, meaning that one allele is dominant and the other allele is recessive. So again, we can go ahead and label these parent number 1 and parent number 2. Alright, so let's go ahead and place these genotypes in their places around this monohybrid cross. So on the top here, we'll do parent number 1, and right here, we'll do parent number 2. For parent number 1, well, we have this genotype of TT, and we know that it's going to be one allele here and one allele here. For the next parent, we have capital T and lowercase t. And so we have those one allele here and the other allele here. Okay, so now it's time to find the offspring's genotype. So for this first box, the vertical part is large T and the horizontal part is large T. For the next one, we have big T and big T. For this one, the vertical part is big T and the horizontal part is little t. For this fourth one, we have the vertical part as large T and the horizontal part as little t. Alright, so these in red are going to be the genotypes for these offspring. So let's find the genotypic ratios. So we have here, remember we're looking at the letters. So we have this one and this one. So we have 2 of these and we have 2 of these. Big T, little t. So we can say that 50% of the offspring are going to be homozygous dominant and the other half are going to be 50% heterozygous. And that's this other type. These are the only genotypes present. We have a homozygous dominant and we have a heterozygous genotype. Now let's find the phenotypic ratios. So we ask ourselves, for this genotype, what will that phenotype be? Well, we know that if we have 2 large T's, the phenotype will be tall. If we have a genotype of capital T and little t, we know that that phenotype will also be tall. Okay, so all of them are going to be tall. Okay, so 100% of the offspring are going to be tall. So for the phenotype, I would recommend looking at each genotype and asking, what phenotype does that give? So how do we know it's tall? Well, we know that if we have at least one dominant allele, that's going to be the phenotype that's going to be expressed. And we know that large T equals tall. Okay, so if you have at least one dominant allele in this problem, you're going to be tall. You would have to have 2 little t's to be short, okay, because that's a recessive allele. Okay, and so the genotype and the phenotype ratios are right here. We have 100% of them are going to be tall, and the genotypes are going to be 50% homozygous dominant and 50% heterozygous. Consider these 2 genes for a special rabbit, and then write the genotypes for each parent. For one allele, the allele for gray eyes, A, is dominant to the allele for black eyes, little a. For another gene, the allele for fluffy ears, capital F is dominant to the allele for smooth ears, little f. Now we have this information. 2 rabbits are crossed. One has black eyes and is heterozygous for fluffy ears. The other rabbit is homozygous dominant for gray eyes and has smooth ears. For this question, we need to find the genotypes of the parents, and we are not going to be doing any monohybrid or dihybrid cross. However, this type of question is very relevant to the dihybrid cross questions, in which we will be dealing with 2 genes. So let's write our notes. We have gene number 1 is going to code for eye color. Okay, and so for eye color, we have capital A is going to be gray eyes, and lowercase a is going to be black eyes. Then we have another gene. Okay, we have this thing for fluffy ears. So we have capital F for fluffy ears, and we have lowercase f for smooth ears. Now we need to find the genotypes. Okay, and so the capital letters are going to be indicating the dominant alleles. Let's do parent number 1. So 1 is black-eyed and heterozygous for fluffy ears. So first of all, black eyes. So to have black eyes, we're looking at this phenotype, and that corresponds with the little a allele. Now the little a allele is recessive, meaning that we need 2 of them to make black eyes. So little a will be the only way to have black eyes. Now we have heterozygous for fluffy ears. Heterozygous means that we have 1 dominant and 1 recessive allele. And now we're talking about the ear texture. Okay, so we're talking about this. So now we have 1 capital F, and the other one is a lowercase f. So this is parent number 1. Now let's do parent number 2. So parent number 2 has a homozygous dominant for gray eyes and smooth ears. Okay, so gray eyes, now we're talking about eye color, and we're talking about homozygous dominant, meaning that both alleles for eye color are going to be dominant, which is the capital A, capital A. Now talking about ear texture, we have smooth ears, and smooth ears is here. And smooth ears correspond to a recessive allele, meaning that we need 2 recessive alleles to have fluffy ears. Okay, so parent number 1 is homozygous recessive for the eye color and heterozygous for the ear texture. Okay, and we have that as little a, capital F, little f. And then for this one, the other parent, we have the homozygous dominant for eye color and homozygous recessive for the ear texture. So capital A, and then little f, little f. Green, capital G, is dominant to red, little g, for flower color. Producing many seeds, capital H, is dominant to producing fewer seeds, little h. A heterozygous green and homozygous recessive seed-producing plant is crossed with a homozygous red and a heterozygous seed-producing plant. Find the resulting ratio of genotypes and phenotypes. So as usual, we're going to write our notes on the side so we can keep track of the alleles and the phenotypes they correspond to. So capital G refers to green, and little g refers to red. Next we have capital H, which is many seeds, and we have lowercase h, which is fewer seeds. Alright, so our next step is to find the genotypes of the parents. So one of the parents is heterozygous green and homozygous recessive for seed production. Okay, so heterozygous for color means that we have one dominant and one recessive allele. So now dealing with color, that is going to be capital G and little g for color. And then we have a homozygous recessive for the seed production. Now we're talking about the H's, okay, these types of alleles. And for these, we have homozygous recessive. So both alleles for seed production need to be recessive. So that means little h, little h. Okay, so this is parent number 1. And let's find the genotype of the other parent. So this is going to be homozygous red and heterozygous for seed production. So if we're talking about this part, homozygous red, well, red indicates we're talking about color. Okay, so we're dealing with the G alleles. And then homozygous red. So to produce red, well, we're talking about this allele. And because little g is recessive, we need 2 of them to have a red color. Alright, so this is going to be the genotype for the color. Now let's do the heterozygous for seed production. So for seed production, we're talking about many or fewer. And now we're talking about H. So heterozygous means that we have one dominant and one recessive. So one capital H and the other one has to be lowercase h for parent number 2. A dihybrid cross is similar to the monohybrid cross. However, we're dealing with 2 genes, so it's going to be slightly different. On the top part, we will have one parent, and on the side part, we will have another parent. Okay, and those will be the genotypes. But in each space, we'll be having 2 alleles. Okay, so instead of just one of them, we'll have 2. And the 2 alleles are going to be coming from the different genes. So G, okay, this is one gene, and the other gene deals with H. So when I say that for, let's say, this part, there's going to be 2 alleles from parent number 1, well, that means that we need to make combinations. So one combination is going to be the big G and little h. Okay? Notice that these alleles are coming from different genes. Next, we have a different combination. So let's do another one. Large G and little h. Okay? And so we're basically taking one of the alleles from one of the genes, so large G, and we're matching that up with the 2 other alleles from the other gene. So with the, the gene for seed production, okay, for H. Okay, so large G is going to meet up with both alleles from the other gene, okay, like this. Now we're going to be doing the same thing for this recessive allele here. Okay, so this one's the little g is going to meet up with this one and this one. So then we take the next allele, and this is going to meet up with the alleles from the other gene. Okay, so the other combination is little g and little h, and then that crosses off this one, and now we have little g and little h for the last one. Now let's find the combinations for this parent number 2. So for parent number 2, same thing. So we'll take the first one and we'll cross it with the first allele of the second gene and the second allele of the second gene. So the first one is little g and capital H. The other one is little g and little h. And then we do the same thing for the second allele. So this one is going to meet up with the first allele of the other gene and the second allele of the second gene. So for the first one we have little g, large H, and then we have the last one is little g and little h. Similar to the one we did for the monohybrid cross, for this first box we're going to see what's the vertical part and what's the horizontal part. Large g and little h. Okay, and then for this horizontal part we have little g and big H. Okay, which we can write like this. I'll be keeping to the order of the capital letter before the lowercase letter, but written in either order it's the same thing. Now for this second box we'll do the same thing. So vertical part, capital G, and then we have this other part, h, little h. And then for the horizontal part we have a lowercase g, and for the other one we have a capital H. Now for this next part we have little g, little h, and then for the horizontal part we have little g and capital H. For this one we have little g and little h, and then for the horizontal part again we have little g and capital H. And we'll just keep doing that for the rest of these. So for this one, again we have the vertical part as large g and little h, and then for the horizontal part we have little g and little h. Okay, and now the vertical part is little g, we have the h here, and then we also have the horizontal part, which is another little g and another little h. Okay, now we're back to the vertical part as big G. The other part of this is going to be little h. You can also think about it as putting the g's together, so the vertical part for g is large G, and the horizontal g is little g. Okay? And then the vertical part for the h is little h, and the horizontal is also little h. So you can just group the letters together. Alright, so this completes the genotypes of the offspring. Now it's time to find the ratios. So we'll find the genotypic ratio first. So basically we're seeing what type of allele combinations we have. So one of the genotypes we have is when we have heterozygous for both genes. So large G, little g, and big H and little h. And we'll highlight the ones that are all like this. We also have another type, and that type is when we have the homozygous recessive for the color and the heterozygous for the seed production. So we'll highlight all the ones with 2 little g's and 1 large H and 1 small h. Next, let's highlight the other type. So this is when we have a heterozygous for the color and homozygous recessive for the seed production. Okay, so let's highlight all the ones with a large G and a little g and the 2 small h's. And lastly, we'll highlight the ones with homozygous recessive for both genes. Okay, so 2 little g's and 2 little h's. Okay, and next we're going to find the ratios. So one of the genotypes we have is big G, little g, big H, little h. And remember, we have total of in here is 16 rectangles. So how many were of this genotype? Well, let's count. We have 4, we're counting the blue. So 1, 2, 3, 4. Okay, so 4 of them are like that. So 25% are like this. Now let's see what other genotypes we have. Let's do the purple one. So the purple one says we have 2 little g's, 1 large H, and 1 small h. So for this genotype, well, we have another 4 of them. So this is 25%. Because 4 over 16, well, if you do your math and convert this fraction to percents, then it's 25%. Now let's do the ones in green. The ones in green say we have large G, and 2 little h's. And there's another 4 of these, so 25% are like this. Now let's do the ones in red. So the ones in red say little g, and 2 little h's. Well, there are 4 of them, so this is also going to be 25%. So all in all, the genotype ratios or percentages should add up to 100, okay? 100%. Because if we're talking about all of them, we're talking about 100% of them. Now we need to find the phenotypic ratios. Okay, so that's looking at the genotypes and asking what type of phenotype will that produce? And we're going to find that for these 2 genes. So we're going to find the color and how many seeds they're going to produce. Okay, fewer or many. Let's start with the one in purple. So large G, large H, and little h. Heterozygous for both genes. So if you're heterozygous for both genes, well, you're going to be, first of all, if you have this one, okay, you're going to be green. Now looking at the other gene, with these alleles, you're going to be producing many seeds. And we know that that percentage is going to be 25%. Now let's do the one in green. So little g, and big H and little h. So this is going to be, let's see. So if we have 2 little g's, well, that's going to produce red. Okay, because little g is going to be the recessive allele. So this is going to be red. And then for how many seeds it's going to produce, well, we have heterozygous for that gene. So that's going to be many seeds. Okay, so let's do this one. So for this one, well, let's see. For this part, we have big G, little g, means that for color, we're going to be green. And I'll put 25% here. There we go. So green and little h. So looking at this part, for this gene, well, we're talking about fewer seeds because we're talking about little h. We know that if we have 2 of them, right, 2 of the recessive alleles will be producing fewer seeds. And that will be 25%. And lastly, we have this one. So homozygous recessive for both genes. Now looking at this first part, we see 2 little g's. So 2 little g's we know is going to be red. And for this next part, little h, well, we're talking again about fewer seeds. And that will be 25% of the offspring. Okay, so these are the genotypic and the phenotypic ratios for this dihybrid cross problem.